of eye. Before learning about embryology of eye, just have a glance on anatomy of eye. Usually, development of eye starts at 22nd day of intrauterine life. Basically, three different tissues are involved in the formation of eye. Those are formation of optic vesicle and optic stalk, lens placode, and the mesenchyme surrounding the optic vesicle. Let's discuss formation of optic vesicle and optic stalk. Optic vesicle and optic stalk develops from neural plate, that is from prosencephalon. Depression of neural plate occurs and there is thickening of prosencephalon on either side, and bulging out of prosencephalon occurs on either side. Bulging results in formation of optic sulcus. This optic sulcus bulges more and forms optic vesicle. After that, fusion of light cells occurs and the optic vesicles get separated from the neural plate. Let's have a brief glance of fourth week embryo showing prosencephalon, mesencephalon, rhombencephalon and spinal cord. This is the lateral view of fourth week embryo. When we see from anteroposterior aspect, we can see the prosencephalon, mesencephalon and rhombencephalon and spinal cord like this. When we cut through prosencephalon along the optic vesicles, we get this section. There occurs a constriction on the proximal aspect of optic vesicles which results in the formation of optic stalk, and two optic vesicles are bulged out portions on either side of the optic stalks. Formation of lens vesicle or lens placode. There is a surface ectoderm overlying the optic vesicle and it is known as lens placode when that surface ectoderm gets thickened. Optic vesicle grows out laterally and results in the thickening of overlying surface ectoderm. It is known as lens placode. We can see here there is outgrowth of optic vesicle which results in thickening of surface ectoderm, and the surface ectoderm bulges inward into the optic vesicle, and optic vesicle forms a optic cup which has two layers, inner layer and outer layer, and the lens placode gets inserted and gets separated from the surface ectoderm by the end of 33rd day of gestation. Here we can see a little deficiency of optic cup on the inferior side known as choroidal or fetal fissure. Let's see changes in associated mesenchyme that is surrounding the optic vesicle. Generally, it forms meninges around the brain, and some part of those mesenchyme surrounds the eye globe. It has two parts, superficial fibrous layer and deeper vascular layer. Fibrous layer corresponds to dura and vascular layer corresponds to pia arachnoid membrane. This superficial fibrous layer forms corneal stroma anteriorly and sclera and extraocular muscles posteriorly, whereas vascular layer invaginates through the choroidal fissure and forms hyaloid system, and then iridopupillary membrane and forms choroid and ciliary body. Here we can clearly appreciate the optic cup with lens and the pia arachnoid membrane and the dura mater with optic stalk intact. Let's see development of various ocular structures such as retina, crystalline lens, cornea, choroid, conjunctiva, etc. At first, let's discuss retina. We know that the optic cup has an outer single layer and inner layer. The outer single layer gives rise to pigment cell layer, and the inner cell layer proliferates and forms nervous cell layers of retina. And they include rods and cones, photoreceptor layer, that is matrix cell layer, and outer limiting membrane, external nuclear layer, external flexiform layer, internal nuclear layer, and internal flexiform layer. These all layers conclude mantle layer of retina, and then ganglion cell layer, optic nerve fiber layer, and internal limiting membrane forms marginal layer. Here, thus retina has 10 layers. These two layers continues anteriorly as single layer and forms part of iris and ciliary body. You can clearly appreciate it from this picture. By the end of 8th month, all retinal layers are histologically seen in the embryo, and then maturation of photoreceptors continues even after birth also. So visual acuity of children increases when the children grows. Crystalline lens. These are formed from surface ectoderm. Already we have seen how lens placoid is formed and then results in formation of lens vesicle. Here it is the early lens vesicle, and there is an elongation of posterior layer of epithelial cells which results in formation of primary lens fibers, and this process takes place up to third month of gestation and also known as embryonic lens. Here is the late lens vesicle with elongated posterior epithelial cells. Space in the lens vesicle, and there is formation of embryonic lens, also known as nucleus. Secondary lens fibers are formed from equatorial part of anterior epithelium of lens, and there is active proliferation of these secondary lens fibers throughout the lifetime of a person. And there is a lens capsule that is present on the anterior aspect of the lens, and this is true basement membrane produced by epithelium present on the external aspect of the lens, and this is the thickest part in the body, thickest membrane in the body. Secondary lens fibers are called as different names at different developmental stages, such as they are called as fetal nucleus during 3rd to 8th month of gestation, infantile nucleus at last weeks of fetal life till puberty, and adult nucleus that is after puberty, and cortex that is superficial lens fibers of adult lens. Next, cornea. Cornea has totally 5 layers, of which epithelium is formed from. Here it is epithelium, and this is formed from surface ectoderm, and the inner stroma and resting descendant layers are formed from fibrous layers of mesenchyme. Here you can appreciate the cornea. We can see the cornea continues posteriorly as sclera, and this is also formed from fibrous layer of mesenchyme. Then choroid. Choroid is formed from inner vascular layer of mesenchyme. Here you can clearly appreciate the choroid that is present in the inner aspect, which is formed from inner vascular layer of mesenchyme. Ciliary body has 2 layers of epithelium and are formed from the anterior part of 2 layers of optic cup which are fused and forms iris also. Stroma, that is ciliary muscle and blood vessels of ciliary body, are formed from vascular layer of mesenchyme. Iris. Iris has 2 layers of epithelium which are formed by iridopupillary membrane from optic cup. Iridopupillary membrane. Next, sphincter and director pupillae are formed from anterior epithelium, whereas stroma and blood vessels are formed from mesenchyme. Vitreous chamber. Based on the developmental stage, it is either primary or primitive during 4 to 5 weeks of gestation, secondary or definitive or vitreous proper during 2 months of gestation, and tertiary vitreous, which is during 3 to 4 months of gestation. Next, eyelids. Eyelids are formed by reduplication of surface ectoderm above and below cornea, and they come closely in contact, and thus closure of eyelids takes place. Separation of eyelids takes place after 7th month of intrauterine life, that is gestation, and conjunctiva is formed from ectoderm, and it totally lines the lids and the globe of eye. Thank you.